Welcome to our webinar about the effect of different SARS-CoV-2 variants on COVID-19 assays and vaccines, organized by Antibodies Online in cooperation with NanoTemper Technologies. My name is Stefan Pellenz, Product Manager at Antibodies Online, and I will be presenting today together with Andreas Langer, Senior R&D Scientist at NanoTemper. The webinar will be recorded, and we will make it available in our website within the next days. Any personal information about participants will be removed, so please don't hesitate to use the chat window to ask any questions that will be answered in the Q&A session subsequently to the presentation. After a few introductory words about today's organizers and speakers, I'm going to talk a little bit about SARS-CoV-2 proteins, and in particular the S protein and its significance in context with existing vaccines. Subsequently, we will take a closer look at the S protein structure and the effects of some of the mutations in virus variants of concern, and I will provide some background on one exemplary S protein antibody. Andreas will then present detailed affinity data that we have produced for this antibody and S proteins from different SARS-CoV-2 variants, as well as for some other antibodies. After putting this data in perspective to some real-world data, we conclude the presentation with some summarizing remarks and the aforementioned Q&A session. Organizers of this webinar are Antibodies Online and NanoTemper Technologies. Antibodies Online was founded in 2006 as a spin-off of the Aachen Technical University. We are located in Aachen in Germany and in Philadelphia in the US. And our mission is to make the search and purchase of antibodies, proteins, kits, and other proteomics products easy and straightforward. We count more than 6,000 institutions in 65 countries as our customers, some of which are indicated here. In April 2020, with 1 month after SARS-CoV-2 had been declared a global pandemic, Antibodies Online offered 600 SARS-CoV-2 related antibodies, proteins, and kits. 1 month later, by May 2020, many of these had already been delivered to customers in over 25 countries. And by now, in June 2021, we have delivered an ever-increasing number of products to more than 470 labs. This confirms Antibodies Online as a trusted source for COVID-19 research. Beside great products, we strive to provide great content relating to the research fields these products are being used in. We have, for example, released the world's first product-centric SARS-CoV-2 mutations tracker to help our customers keep pace with a highly dynamic COVID-19 research. We have also compiled a SARS-CoV-2 handbook providing an overview of SARS-CoV-2 virus, COVID-19 biology, and related products. The mutations tracker and the handbook are both freely accessible on antibodiesonline.com. Validation data plays a key role at Antibodies Online, and we pick and recommend the products that come with the best data. We do also generate additional data through our independent validation program. The data we will present in this webinar fits into our data mantra as we go beyond what you usually get to see. NanoTemper Technologies, on the other hand, was founded in 2008 as a spin-off of Ludwig Maximilians University in Munich. The company is headquartered in Munich, Germany, and has by now 8 additional offices in Europe, the US, China, India, and Japan, with a total of approximately 160 employees. The product portfolio comprises 4 different product lines for the characterization of proteins. The monolith and the Ianthus platforms, which use the effects of microscale thermophoresis and temperature-related intensity change to measure molecular interactions, and the Prometheus and Tycho platforms, which apply nanodifferential scanning fluorimetry to measure protein stability. All in all, more than 1,000 NanoTemper instruments are used all around the world and were cited in more than 5,000 publications. Our speakers today are Drs. Andreas Langer and me, Stefan Pellenz. Andreas holds a doctorate in biophysics from the Technical University of Munich and has extensive experience in protein interactions. As application scientist, Andreas had advised NanoTemper Technologies distributors in Japan, India, and South America on the use of the different NanoTemper instrument platforms. He is now R&D Scientist at NanoTemper Technologies. I have obtained a PhD in genetics from University Paris 7, followed by a postdoc at the University of Washington in Seattle, Washington. Both during my PhD and postdoc, I have worked with engineered DNA and nucleases, and I have therefore a long-standing interest in protein structure and how the structure influences protein function. I am now working as a product manager at Antibodies Online, and I'm responsible for our independent validation program. So after these initial words, this brings us to the main topic of today's presentation. SARS-CoV-2 proteins. SARS-CoV-2, the causative agent of COVID-19, has a positive sense single-strand RNA genome with a size of almost 30 kilobases. This genome encodes a total of 26 proteins, 16 of which are so-called non-structural proteins, or NSPs. That means they do not contribute to the structure of the assembled virion. Their function is primarily replication and the production of new virus once SARS-CoV-2 enters a host cell. Among these are the two proteases, PL-Pro and the main protease 3CL-Pro, and the RNA-dependent polymerase and the exonuclease for proofreading during genome replication, the main reason for the comparatively low mutation frequency of SARS-CoV-2. In addition, the SARS-CoV-2 genome contains sequences for 9 accessory factors whose function in virus replication, modulation of the host's immune response, or packaging remain largely unclear. And then lastly, the genome contains 4 structural proteins, the spike, envelope, membrane, and nucleocapsid protein that make up the assembled virion. I will today be focusing on the spike protein shown here in orange. So why the focus on the spike protein? Shown here is a list of SARS-CoV-2 vaccines that have been approved until now for use in humans. They rely on different technologies. The BioNTech, Pfizer, and Moderna vaccines, for example, build on an RNA encoding a viral protein packaged in lipid particles. Other vaccines, like those from AstraZeneca, the Sputnik V vaccine, Johnson & Johnson's Janssen, and the CanSino Biologics vaccine, rely on delivery of viral S protein DNA and subsequent production of the antigen itself by the infected cell. While the delivery of the genetic information for the antigen varies between these vaccines, they all rely solely on the S protein to mount an immune response against SARS-CoV-2. More recently, the Novavax vaccine has become the first approved protein-based formulation, and here as well, the S protein is the pertinent antigen. The two vaccines shown here on the bottom 2 rows of the table, the Sinovac Biotech vaccine and the Indian Covaxin, use inactivated SARS-CoV-2 virions. Therefore, they display also the other structural proteins as opposed to only the S protein. However, the S protein appears to be the most immunogenic of the SARS-CoV-2 structural proteins, and one can safely assume that even in these whole virion vaccines, the S protein plays a crucial role. The S protein is also the body's first point of contact with the SARS-CoV-2 virus. So in short, the S protein is very important for all SARS-CoV-2 vaccines, independent of the selected platform. In order to better understand the body's interaction with SARS-CoV-2, it is helpful to have a closer look at the S protein structure. The S protein protrudes from the spherical virion, shown here on the left, and gives it its characteristic appearance, resembling a crown that has earned the group of coronaviruses its name. Zooming in on the S protein here on the right, well, middle and right, shows that it's actually a homotrimeric complex consisting of 3 monomers, shown here in yellow, gray, and green. The lower S2 section of the complex contains the trimerization interface and the membrane proximal stalk that's important for membrane fusion and virus entry. The upper S1 section contains an N-terminal domain, or NTD, and the receptor binding domain, or RBD. The 3 RBDs have to transition from a closed conformation, shown here in the middle, to an open position, shown here for one of the RBDs on the right, for the virus to contact the ACE2 receptor on host cells. While the NTD has been shown to contain many immunodominant regions eliciting a T-cell response, the RBD appears to be the primary target for antibodies. So therefore, mutations within the RBD are highly relevant for the effectiveness of antibodies, either in the host's immune response or in immunoassays. Having a closer look at the S protein mutations present in SARS-CoV-2 variants of concern and variants of interest, shown here on the left, revealed several amino acid changes that can potentially affect binding of antibodies, recognizing the canonical RBD section, again shown here in orange. Especially the mutation E484K stands out because an acidic negatively charged aspartate has been changed to a basic positively charged glycine. Biochemically, this is a significant change, and this is consistent with the observation that variants containing this mutation, like for example B1351 beta and P1 delta, reduce the effectiveness of treatment with serum from convalescent patients and some of the existing vaccines. It is also to be expected that some of these mutations might negatively influence the function of some existing antibodies used for SARS-CoV-2 lateral flow assays, ELISAs, and other immunoassays. In order to verify the usability of any antibodies targeting the S protein, one has therefore to test if they retain the ability to bind the intended target in novel virus lineages. So shown here from left to right are structures from SARS-CoV-2 variants of concern B117 alpha, B1351 beta, P1 gamma, and B16172 delta. For clarity's sake, amino acids that differ from the S protein of the canonical Wuhan wild-type strain, these are indicated here only for the gray monomer, but obviously they're present also in the green and the yellow monomer. The attentive observer will remark that many of the SARS-CoV-2 variants of concern appearing in the daily news are located within the S proteins RBD, so in this region here. These are, for example, E484K, this one here, for example, in the beta and gamma variants, or L452R and T478K in the delta variant. This is actually why the delta variant has initially been called double mutation as well, because of these two. It's consistent with the observation that S proteins from variants that are more readily spread have an increased propensity for the RBD in the upstate and show increased ACE2 receptor binding because of these mutations actually. So additionally, mutations like E484K have local destabilizing effects that can negatively impact binding of neutralizing antibodies. Production of neutralizing antibodies is actually one aspect of immunity upon vaccination or infection with SARS-CoV-2. Simply put, a neutralizing antibody gets in the way of the S protein's interaction with the ACE2 receptor on the surface of the host cell. The main target for neutralizing antibody is, as I have mentioned before, the S protein RBD, shown here on the right in more detail. Depending on the location of the target epitopes, neutralizing antibodies are classified into classes 1, 2, or 3. An antibody attached to these areas of the RBD interferes with binding of the ACE2 host cell receptors. This can be sterically or also by directly blocking residues. Of particular interest are the class 2 antibodies, indicated here in yellow. Because these antibodies actively block the receptor binding motif, or the RBM, which is kind of in this area here. This motif contains amino acids that form direct contacts with residues on ACE2. Here one finds, for example, the residues 484, and it has been shown that the E484K mutation effectively abolishes binding of class 2 neutralizing antibodies, thus negating their neutralizing effects. Another position interesting in context with the Delta variant is the mutation in amino acid 452. This sounds as if it's actually kind of far away from the position 484. The funny thing is, if one has a closer look at the structure, those two end up being pretty much right next to each other, the two residues. So it makes sense that both of them would have a similar effect. So in contrast to class 1, 2, and 3 antibodies, there's a fourth class of S protein antibodies, indicated here in red. These class 4 antibodies are characterized by the fact that they bind in none of the three other sites of the other classes. They do also not contact any residues that bind directly to ACE2. One of these antibodies is the neutralizing antibody CR3022, indicated here. The antibody CR3022 was originally isolated from the serum of a convalescent SARS-CoV-2 patient about 15 years ago. The antibody on its own does not show strong neutralizing effect, since it binds to a conserved epitope that does not interfere with ACE2 binding, which is what I had just shown in on the slide before. It does, however, synergize well with other neutralizing antibodies. That means the combined effect of the two neutralizing antibodies is larger than would be expected. Besides its function as a neutralizing antibody, CR3022 is frequently used as a reference in immunoassays targeting the SARS-CoV-2 S protein across different variants. So shown here on the right is a schematic representation of the antibody's epitopes, here in yellow, indicated here with the amino acids. Within the S proteins RBD, again shown in orange here. Mutations in different variants are indicated in the rows above for each variant of concern in gray or variant of interest here in green. Based on the RBD's primary sequence, the epitopes of antibody CR3022 do not include any of the amino acid mutations in these variants. I mean, there's, or simply speaking, in this representation, there's no overlap between these orange blocks and the yellow blocks. So unless the quaternary structure of the variant's S protein trimer has any effect on binding of CR3022, we hypothesize that the antibody still binds to all variants shown here, which includes pretty much all the variants that you find also covered in the daily press, including the Delta variant here. So in order to test this hypothesis, we sourced high-quality, full-length trimeric SARS-CoV-2 S proteins, including the membrane proximal stalk from our protein expression lab here in Germany. S proteins were tested were those of the canonical Wuhan wild-type strain here, that we will refer to in the following as just as wild-type, and S proteins from different variants, B117 alpha, B1 351 beta, P1 gamma, B1 429 epsilon, B1 525 eta, and B1 6171 kappa. We did not test the Delta variant, this already as a disclaimer beforehand, because when these proteins were ordered, there was no agreed upon sequence for the Delta variant available yet. So at this point it was just the Indian variant, and this is pretty much what comes down now to the Kappa variant. So for the measurements, we teamed up with NanoTemper Technologies, and Andreas will show you on the next slides whether our hypothesis regarding binding of different S proteins variant by antibody CR3022 held true. Here you go, Andreas.
Yeah, thank you, Stefan. So our aim at NanoTemper was to really quantify the strength of the interaction between this CR3022 antibody and these 7 S protein variants that we received. And to do this, we used our monolith instrument that you can see in this picture here, which is using microscale thermophoresis technology and can be used to measure binding affinities between 2 molecules. So we get basically the dissociation constant, or the KD parameter, that describes how strong 2 molecules interact. When you want to do an MST measurement, you have to fluorescently label one of your 2 binding partners. And here in this constellation, it made a lot of sense for us to fluorescently label this CR3022 antibody, and then we could just use these 7 different variants as ligands and could get very comparable data. Now, the first step when you want to do these measurements is you have to prepare a dilution series of your ligand. So here in the S protein, and we chose a range between 7.6 picomolar and 250 nanomolar. So this higher concentration here allowed us that we could also see binding of some weaker binding variants, if there would be any. Then as a next step, this dilution series gets mixed with the fluorescently labeled antibody. And we had here in the assay a final concentration of only 2.5 nanomolar. So we can go down so low because it's a fluorescence-based method. So you can really decrease the concentration. You don't use a lot of sample. And it also allows you to measure really strong affinities, even in the picomolar KD range. And here I want to show you already the results of these measurements. What you can see here are dose response curves. We have at the x-axis the concentration of the S protein, and on the y-axis it's the so-called F-norm value, which is the signal that you measure with the monolith instrument. And since we are interested in comparing these different variants, it makes sense to look at the normalized display. This is called the fraction bound plot. And then we get these dose response curves that start at 0 and end at 1. And basically, the more they are shifted to the right, it means the weaker is the interaction. The more they are on the left, the stronger is the interaction. And from this, I think we can already see a lot what's going on. The first one we have here, the red one, that's the wild type S protein. And this gave us the strongest binding interaction. So we measured here a KD value of around 1 nanomolar. And that's a really strong binding. So antibodies that bind in a single-digit nanomolar range, they're really good antibodies. A lot of commercially available antibodies have much higher KDs, maybe even micromolar. That's a factor of 1,000 weaker binding. So 1 nanomolar is really, really good. If you have an even better antibody, you might sometimes even have picomolar affinities. So for example, 3-digit picomolar KDs can also be observed. The next we could see was that the B117, the alpha variant, showed a very similar binding. So apparently this mutation that the B117 has does not affect much how this antibody binds to the S protein. But then we already saw some differences. So for example, the P1 variant here showed a factor 3 weaker binding. And then there was an even larger gap, and we had 3 more variants that showed a factor of 10 to 20 weaker binding. And that was the B1351. So the variant that originated in South Africa, and also the B1617.1, the Kappa variant from India originally, and also the B1429. And that was very interesting because these two here share this mutation at the position 452. And that's also the mutation that the Delta variant has. So one could now guess that the Delta variant might also show a weaker binding. But all in all, we could observe that still this antibody bound to all 7 variants, because a KD of 20 nanomolar, that was the worst one here, is still a pretty good KD value. So this antibody is totally perfect to be used in, for example, ELISA or in neutralization assays. For comparison, we also have here the KD value between the S protein and the host of COVID-19, the ACE2 receptor. And that was found to be around 15 nanomolar. So you can see it's all in the similar ranges. One could now maybe speculate that these more infectious variants, the Delta variant, maybe have some higher affinities. So then it could also come down to a lower nanomolar KD. Now, seeing these differences with the CR3022, we are very curious to see if maybe also other antibodies are affected by the variants. Therefore, we tested 2 more antibodies that were specifically raised against the SARS-CoV-2 S protein, the S1 subunit. So they were supposed to bind even stronger, and we could confirm this. So the SP422 that we tested here showed even a picomolar affinity against the wild type and also against the B117. So both here have maybe a KD between 500 picomolar and 1 nanomolar. The other variants we tested, the B1351 and the P1 and the B16171, showed a slightly weaker binding. So again, here it's still a quite good affinity, but there is a difference between these 2 groups. And then to our surprise, the second one we tested, the SP185 antibody, again showed picomolar affinity to the wild type and the B117. So these curves are perfectly overlaying, but then for the latter 3 variants here, we didn't see binding at all. So maybe at the very highest concentration, there was some onset. You would need here much higher concentrations to get the correct, the KD value here. Probably it's around 1 micromolar, but in the concentration range we tested, there was no binding at all. So apparently here, these amino acid substitutions, they really can abolish completely the binding of this antibody. At this point, you might be asking, how is MST different, the MST technology, to other techniques that maybe you are using your own on testing antibody-antigen interactions? And for this, I prepared this small table to tell you a bit about the similarities between these methods and the differences. Here I compare MST with the SPR technology that is routinely used to measure antigen binding, and also the ELISA method. So let's start with a similarity, and that is the approach. So in all these three methods, we are following a titration approach. This means you want to test if two molecules interact, and you do a dilution series of one of these molecules, and then you check what concentrations do you need to see some binding. Now, the first difference is the readout. So the MST technology and also the SPR technology, they are quantitative methods. This means that you get really a hard number as a result. You get a KD value, and this KD value tells you how strong these two molecules interact. And you can use this value also to compare different antibodies, for example. ELISA is only a semi-quantitative method, which means you don't get a KD. You can still see if two molecules are interacting, but it's more difficult to compare because of this enzymatic readout at the end. Regarding immobilization, it's another difference. MST is a method that works completely in solution. So both molecules are in solution. It's fluorescence based. So we can do this because one of these two molecules is fluorescent. In SPR and in ELISA, we have an immobilization. So one of these two molecules needs to be immobilized on the surface. This can be done in SPR through different immobilization chemistries. So if your protein has a certain tag, for example, you can use that to immobilize it even site-specifically. In ELISA, it's often a non-specific immobilization. So here the protein absorbs non-specifically to plastic, and you have to be aware that not all proteins might be happy with that. So it could happen that the protein is perhaps denaturing when it's absorbing, and then you don't have binding anymore. A second difference is fluidics. Again, MST is an in-solution measurement. We measure in small glass capillaries, so there's no need for fluidics. In SPR and in ELISA, you have to have a way to exchange the solution on top of your immobilized protein. And in SPR, this is done by a flow cell, by a fluidic system. In ELISA, you do this with multiple washing cycles. And this is another drawback of ELISA, because if you have a weak binder, it might be that while you are washing, it already dissociates, it goes off, and then you will think it didn't bind in the beginning, although maybe it was bound before. So this is something that does not happen with MST and SPR. Here, you can even measure very weak affinities. For example, if you have a micromolar binder, you will still be able to get a KD value for that. Regarding sample consumption, that's a point that I personally found very beneficial. So when I did these measurements, I only needed about 10 microliters per concentration. And especially if you have these different spike variants, the S protein variants, you don't have a lot of volume, a lot of sample to work with. You have to be able to work with a few micrograms, especially if you want to test a lot of different antibodies. And that comes in really handy if you only need a small volume instead of, for example, a factor of 5 higher volume. So it means you can do 5 more different antibodies than with some other technique. And finally, regarding time to result, in MST, I told you you have to do a labeling in the beginning that takes about 1 hour. In this case, it was the labeling of the antibody. And once you have done this, you can prepare aliquots. You can store them at -80 degrees. And then in this case, what I did was whenever I received a new S protein variant from Antibodies Online, I could take out a fresh antibody and I could do my titration measurement and obtain the KD for the interaction. In SPR, it might take maybe a little bit longer. In ELISA, that's probably the most time-consuming method because you have this many steps of incubation, of washing, of surface blocking. So it can easily take maybe around 3 hours. Yeah, speaking about ELISA, what was very nice in the end was that we could also compare our results from MST with ELISA because Antibodies Online performed also some experiments with 2 of these antibodies, the SP422 and the SP185. And as you can see from this picture here, we have MST in the left column, ELISA on the right. We get kind of the same picture. So we can see the same things happening. Both bind in the same concentration range. We see here this difference between the P1 and the B1351, that it binds a bit weaker. Also in ELISA, it binds a bit weaker. And we also could see that the SP185 antibody here did not bind at all these variants of concern. And I personally find this very remarkable because also the orientation was very different in these two methods. So in MST, we had a labeled antibody and we titrated the S protein, and in ELISA, the S protein was actually immobilized and the antibody was titrated. And if you get the same result doing this completely different orientations, that's a very strong proof that what you see is real and significant, and that also these differences in KD that we measure, that they're really meaningful. In the end, I want to also mention that with MST, you can do also measurements under more challenging conditions. So all this data was collected at 25 degrees and in a buffer solution, but it's also possible to, for example, measure at a higher temperature. So we did some tests at 39 degrees, also in a solution, 50% buffer and 50% human serum or human plasma. And you can still see that this antibody binds under these conditions. So I think it's really cool that you can test all kind of different challenging conditions and get this information. This may be more difficult in other techniques. In techniques with a flow cell, for example, it might be that the fluidic system gets clogged if you have some more complex samples. And in ELISA, the higher temperatures might have some effects on the enzymatic readout. So having you presented these affinity results, I want to give back the word to Stefan, who will now tell you what this means in comparison to real-world data.
Great, thanks, Andreas. So our primary interest for the experiments, just to recall, was to find out whether antibodies used for immunoassays can still bind S proteins of novel SARS-CoV-2 variants. Naturally, this question is also highly relevant for antibodies produced in response to SARS-CoV-2 infection or following a vaccination. So a quick comparison of some real-world data shows that the effect of the S protein mutations is indeed measurable. So there's this first study here on the top from Israel, in which the authors came to the conclusion that the variant of concern B1351 does indeed escape neutralization by class 1 and class 2 antibodies. And in the second publication from South Africa, the BioNTech Pfizer mRNA vaccine was shown to efficiently neutralize SARS-CoV-2 wild type as well as the B117 alpha variant. However, in comparison, neutralization was reduced almost by 7-fold with the B1351 beta variant. There's actually more data since then, but just as an exemplary data set, I thought I'd leave it at this. So it is still not clear what level of antibody production can be considered a good response to a SARS-CoV-2 infection or vaccination. A decrease in neutralization does not necessarily equal a failed immune response. In addition, antibody production is not the immune system's only means to counter an infection, and there are indications that, for example, the BioNTech Pfizer vaccine provides protection from the novel variants such as the Delta variant. However, it is notable that depending on the antibodies produced, mutations in the S protein can really have significant effects. So that was one too far. That was one too far to the other direction. I'm really sorry for that. So in summary, based on the MST measurements, neutralization antibody CR3022 recognizes the SARS-CoV-2 wild type as well as S proteins from the alpha, beta, gamma, epsilon, eta, and kappa variants. This means that although the binding affinity of SARS-CoV-2 antibodies to the antigens may be affected by mutations, they can remain valuable tools also for novel virus variants depending on the epitopes they bind to. We are now extending these binding measurements to S proteins from other SARS-CoV-2 variants, in particular, obviously, the B16172 Delta variant. Besides, we are already testing additional monoclonal and polyclonal antibodies, as well as different antibody platforms in comparison. So then, yeah, more generally, we at Antibodies Online are constantly extending our offering of SARS-CoV-2 related antibodies, proteins, and assay kits. So if you need, please let us know. You can get in touch with us as indicated here. Both from Europe and US, and that's true both for NanoTemper as well as Antibodies Online. We would like to thank Dr. Susanna Matamoros from the Forschungszentrum Jülich for her help to edit the graphs that we had presented here as well. And in case of any questions or interest in any of the products mentioned here or beyond, please don't hesitate to get in touch, or feel free also to ask any questions in the chat window.